Hello, my name is Joshua Mugabe. I welcome you to Embryonic Development. What is the cell division process directly related to embryonic growth? Embryonic growth depends directly on mitosis. Through this type of cell division, the zygote divides itself, giving birth to a series of cells that by mitosis also compose differentiated tissues and organs until the formation of the complete individual. What is the function of the vitreous in the vertebrate egg? How are these eggs classified according to the amount of vitreous within them? Vitreous, the yolk, is the nutritive material that accumulates the cytoplasm of the egg or the zygote with the function of nourishing the embryo. According to the amount of vitreous in them, the vertebrate egg is classified as oligolecithal, meaning little yolk, centrolecithal, or heterolecithal, having more yolk diffusely distributed, and telolecithal, more yolk concentrated in one end of the egg. What are the animal pole and the vegetal pole of vertebrate egg? The animal pole of a telolecithal egg is the portion of the egg with little vitreous. It is opposite to the vegetal pole, which is the region where the yolk is concentrated. What are the four initial stages of the embryonic development? The four initial stages of the embryonic development are the molar stage, blastula stage, gastrula stage, and the neurular stage. What is the cell division during the first stage of the embryonic development? How is this stage characterized? The cellular division in the first stage of the embryonic development is called cleavage or segmentation. In this stage, several mitoses occur from the zygote, forming the new embryo. What are the cells produced in the first stage of the embryonic stage called? The cells that result from cleavage, the first stage of the embryonic development, are called blastomeres. In this stage, the embryo is called modular, similar to morus, mulberry. After the modular stage, what is the next stage? What is the morphological feature that defines this stage? After passing the modular stage, in which the embryo is compact mass of cells, the next stage is the blastula stage. In the blastula stage, the compactness is lost and the internal cavity is filled with fluid, appears inside the blastocell. After the blastula stage, what is the following stage of the embryonic development? What is the passage from blastula to the next stage called? The blastula turns into the gastrula, a process known as gastrulation. What is gastrulation? How during gastrulation are the first two germ layers formed? What are these germ layers? Gastrulation is the process through which a portion of the blastula wall undergoes invagination inside the blastocells, forming a tube called ancestron or the primitive intestine. The cells of the inner side of the tube form the endoderm, germ layer of the cells of the outer side form the ectoderm, another germ layer. It is the beginning of tissue differentiation in embryonic development. What are the ancestron and the blastophora? What is the stage of the embryonic development in which these structures are formed? What is the destination of the ancestrons and of the blastophora? Ancestron is the tube formed during gastrulation, meaning of invagination of the blastula wall inside the blastocell. It is the origin of the gastrointestinal tract. Blastopore is the opening of the ancestron to the exterior. The blastopore gives birth to one of the extremes or extremities of the digestive tube. The mouth in the protostomes being, or the anus in the deuterostome beings. How is the mesoderm, the third germinal layer of trypoblast animal, formed? The mesoderm appears from differentiation of the endodermal cells that cover the dorsal region of the archetrons. What are the three types of germ layers that form tissues and organs in animals? The three germ layers of the ectoderm, the mesoderm, and the endoderm. How are animals classified according to the germ layers present in their embryonic development? Nidalia are diploblastic, that is, they present only the endoderm and the ectoderm, with the exception of polypheran. All remaining animals are triploblastic. Polypheran do not present differentiated tissues organization, so they don't classify regarding to the germ layer, although sometimes they are mentioned as diploblastic. How do embryos turn from gastrura into neurura? How is the neurura tube formed? What is the embryonic origin of the nervous system in vertebrates? The neurura stage is characterized by the appearance of the neurura tube along the dorsal region of the embryo. The growth of mesoderm in the region induces the differentiation of the ectoderm cells just above. These cells then differentiate, forming the neurotube, the origin of the nervous system is the ectoderm, the same germ layer that gives birth to the skin. What is the notochord? How is this structure formed? The notochord is a rod-like structure that forms the supporting axis of the embryo, giving birth to the vertebral column in the vertebrates. It is formed by differentiation of the mesoderm cells. What is the coeron? To which structures do the coelom give birth? Are all animal coelemates? Coeloms are cavities delimited by the mesoderm. Coelom originates the cavities where the internal organs of the body are located, like the pericardial cavity, peritoneal cavity, and the pleural cavity. Beside the coelomate animals, there are coelomate animals, like the platyhelminths, the pseudocoelomate animals, like the nematodes. What is the germ layer from which coelom originates? The coelom are originated from the mesoderm. What are pleural, pericardium, peritoneum? Pleura is the membrane that covers the lungs and the inner wall of the chest. Pericardium is a membrane that covers the heart. Peritoneum is a membrane that covers most organs of the gastrointestinal tract and part of the abdominal cavity. All these membranes delimits coelom, internal cavities. After neurulema stage and from its ventral portion to the dorsal, how can the morphology of the embryo be classified? The schematic longitudinal section of the embryo after neurulema stage. The outermost layer of cells is the ectoderm. In the ventral region comes the acetoron tube, formed of the endodermal cells. In both sides of the embryo coelom, delimited by the mesoderm, are present. In the central region above the acetoron and in the middle of the coelom, there is the notochord. In the dorsal region just above the notochord lies the neural tube. What are somites? Somites are differentiated portion of the mesodermal tissue, longitudinally distributed along the embryo. Somites originate the muscle tissue and portion of the connective tissues. What are histogenesis and organogenesis? Histogenesis is the process of tissue formation. in the embryonic development. Organogenesis is the process of organ formation before histogenesis and organogenesis. The primitive embryonic structure have been already formed. Germ layers, neurotube, notochord, chorellon, somites. From which germ layer do the epidermis and the nervous system originate? What other organs and tissues made from that germ layer? Epidermis and nervous system are some of the embryonic origin, the ectoderm. The epidermal appendages like the nail, hair, sweat glands, sebaceous glands, the mammalian glands, the end of hypophysis, the cornea, the crystalline lens, and the retina are also derived from the ectoderm. From which germ layer do the blood cells originate? What are the organs and tissue made from that germ layer? Blood cells have a mesodermal embryonic origin. Other organs made from mesoderm are the covering cellus membrane, like the pericardium, peritoneum, and pleura. Muscles, cartilages, dermis, epidermal tissues, kidneys, ureter, bladder, urethra, gonads, blood, and lymph vessel bones. From which germ layer do the liver and the pancreatic originate? What other organs and tissues made from that germ layer? The liver, the pancreas, are originated from the endoderm. Also from the endodermal origin are the epithelial of the airways, epithelial of the bladder, urethra, and the gastrointestinal tube, except of the mouth and anus, alveolar cells of the lungs, thyroid and parathyroid glands. What are twins? Genetically, what are the two types of twins that can be generated? Twins are simultaneously generated within the mother's uterus, offsprings. Twins classified according to the zygosity as monozygotic or as dizygotic twins. Monozygotic twins, also known as identical twins, are those originated from one single fertilized ovum, therefore from one single zygote. Monozygotic twins are genetically identical. That is to say, they have identical genotypes and are necessarily of the same sex. Digozygotic twins, also known as fraternal twins, are those generated from two different ova, fecundated by two different sperm cells. So they are not genetically identical and they are not necessarily of the same sex. What is polyembryon? Polyembryon is a phenomenon in which a single embryo in its embryonic stage divides itself and forming many new individuals of the same sex and genetically identical. This is the way, for example, in which reproduction takes place in armadillos of the genus Dispas. Polyembryon is an example of natural cloning. Thank you very much for participating in embryonic development. Please do like, share, and subscribe.